We're going to shift gears a little bit in this part of Chapter 5 and start to look at Kingdom Protista. And Kingdom Protista is what I refer to as, it used to be the junk drawer for the eukaryotic organisms. If you had organisms out there that didn't fit in with the plants or the animals, then you threw them into Kingdom Protista because they were eukaryotic organisms. And remember that idea, the modern taxonomic or phylogenetic thing, when we're looking at this sort of tree right here, is that we've got a whole bunch of different kingdoms. So this junk drawer is now being divided up into all these different organisms. And if we look at this sort of depiction over here showing this sort of tree of life, we can see this vision on how you had your ancestral prokaryotic cells that came about from the earliest cells that are out there, and then those diversified. And if we look at this as our kingdom Protista that led to these other kingdoms of eukaryotic organisms, but this is where we're running into problems with the classification. And part of the problem is when you look at this, there's actually two very distinctly different groups of organisms that are out here. On this side, we have what are known as the Protozoans and also the slime molds, and these are animal-like eukaryotic cells. They don't have cell walls, and they have the ability to ingest their food, even though they are single-celled organisms that are out there. So they think these are the precursors to the fungi and the animal kingdom that are out there. But also grouped within this kingdom, you have the algae. They're photosynthetic eukaryotes that do have cell walls that are presumed to be the ancestors of the plant kingdom right there. So how do you group plants and animals together? Well, that's the problem when we look at the kingdom Protista. And not only that, but look at this last little thing. Some of these protists that are out there are photosynthetic and they are animal-like. They are plants and animals depending on if it's daytime and if there is food available. And some of the common names that we hear that are out there, the phytoplankton, these are referring to the algae that are out there. And the zooplankton, these are referring to the animal-like ones that are out there. So we've got both the plant-like and the animal-like protozoan or protista that are out there. If we look at them, they do have some really beautiful kinds of structures. And if you look on this one, you can actually see this tough, flexible layer of protein that is around the cell membrane here, because in this organism, there is no cell wall, but they still need to adjust their osmotic balance so that they don't lyse, and that's why we have cell walls. That prevents the osmotic pressure from filling up the cytoplasm to the point where the cell explodes. And when you look at these protozoans, they are in a watery environment, so they need to account for osmotic pressure. So they make this pellicle, which you can almost think of it as like a skin for a single cell there. You can also see in this beautiful differential interference contrast micrograph that I found out there, a contractile vacuole here. And this thing is basically going to be a membrane sac that fills up with water, and then it's got little cytoskeleton elements surrounding it, and they will squeeze this contractile vacuole there, and that's going to cause it to expel water and possibly waste materials. So effectively, this is like a bladder for a single cell that is out there. So really kind of interesting when you look at these kinds of single-celled organisms, that they do have things that we find in multicellular organisms that are out there. We can also find that some of them will actually have protective skeletons or shells around the outside of the body of that cell. And here you can see ones that are actually made of glass, and they're very beautiful kinds of structures. And we can also find ones that are out there that have these shells that are made of calcium carbonate. And then down here you can see a really interesting kind of structure. It's known as a trichocyst. So when you look at that, trick, think hair, and so this is literally saying hairy cell. And this is a protein structure that's going to be buried, sort of embedded in the membrane right there. And when something comes up and disturbs this cell, they're going to send out these trichocysts right here. And that will push the cell away. But also, if you have seen some of those movies of the little blowfish that's out there, it inflates itself so it has these spikes on the outside. That's the same thing that we're seeing here with a single cell. It makes these trichocysts extend out because now the cell is bigger and it's less likely to fit into the mouth of another cell that is out there trying to eat it. So when we look at these guys, they have some really interesting kinds of structures. We can also find cytostomes that they will use for phagocytosis. And also, when you look at these, some of these kinds of organisms that are out there can eat their food whole. So imagine this thing can easily fit bacteria because this is on the order of 200 micrometers in size. With bacteria, you're looking at 1 to 5 micrometers. So they can actually fit a bacteria through their cytostome to get it inside, and they're effectively using the process of phagocytosis. You can also find these are saprobes, like with the fungus kingdom, where they're going to be ingesting these small food molecules that have been digested or broken down previously. When you look in the protozoan category, this is where we see some amazing stuff because these guys are motile. You can also find motility when you look at the algae that are out there, but the algae don't have cilia, and they're not going to necessarily move with the pseudopods, but they will have flagella to swim around in the ocean. And so we can look at some of these structures, and these were used to classify the protozoans that were out there, but now we are using that more reliable genetic comparisons between these things that are out there. So really kind of interesting when we look at the size and shapes and structures on the members of this kingdom Protista, they're all over the place in terms of what they have available, but really kind of interesting to look at under the microscope. Again, we do want to take a look at reproduction because that is a primary feature of life that's out there. And you can find both asexual and sexual reproduction. If it's asexual, you go through that process of mitosis, and it could be something that looks similar to what you would find with a bacteria with binary fission, but it is mitosis. You can also find budding like we saw with the yeast cells, but there is this other weird process that we find in the Protists that's known as schizogony. And it's literally crazy cell division because the nucleus divides over and over and over, and you end up with what's known as a schizon. And so if you look at this cell right here, it's one cell with multiple nuclei inside of it right here. And then the cell can finish off the process of mitosis by doing cytokinesis, and that is where the cytoplasm separates. So this one cell over here divided multiple times, but it stalled the process of mitosis and saved cytokinesis for later on. And now you've got all of these cells coming out of this one cell that finishes the process of mitosis. So it is crazy cell division. And this is how something like malaria can be transmitted so easily. You get infected with one cell, it infects cells in your liver where it goes through schizogony, and then that one cell in your liver or the one cell that infected your liver is going to divide, and it pops your liver cell to release all of these little guys that can then go on to repeat the process there. When we look at sexual reproduction, again, it could be through this process of meiosis, and also there's a video I put out there that shows you a process known as conjugation, which is basically sexual reproduction where two of these cells can actually fuse together. So that plasmogamy that we saw before when we looked at the fungi that are out there. And here is just an example of paramecia that can do this process of conjugation. And I'm not concerned about the details, but I did just want to show you that with paramecia, they actually do have what's known as a macro and a micronucleus here. And so if you look at this, you can see the micronuclei are the ones that are diploid in here. And so they will actually have an exchange of DNA between the micronuclei of these two cells right here. And so if you look at this thing right here, you can see they've actually swapped their nuclei between the cells. And so there is that idea of fertilization that we think about with sperm and egg fusing together. But here there is actually the exchange of the micronuclei between the cells. And then those guys can go through this process of finishing off to restore back to over here. So really kind of an interesting thing that is a sexual exchange of DNA between these cells that are actually getting recombinant kids at the end of this. And so it's really kind of weird to think about that. You can actually transmit this whole nucleus of characteristics rather than just the individuals, and you are going to see that crossing over occurring between these nuclei right here. The other interesting feature that we can see when we look at a lot of members out there, and here we can see an example with protozoans, but we can also see this with the algae that are out there, is these organisms are often going to be growing in an environment that dries out. And so they had to come up with a mechanism to allow them to survive through that. And their mechanism is reminiscent of what we see with the bacteria and the fungi that are out there. They can go into this dormant cyst stage that we can see right here with this protective coating around the outside. And the cells that are inside of the cyst right here are basically sleeping. They are not active, and they will hang out in this dormant cyst stage until the conditions are more favorable. And this is what we'll often see with the protozoans that cause diseases in us, is we ingest the cyst form of it, and then we actually digest off the outer covering here, and that releases this thing into our intestines where there's digested food. And now you've got the trophozoite stage of their life cycle. And when they're in the trophozoite stage, this is when they're actively growing, they're feeding, and they're reproducing. So if you have this thing in your intestines, they are actually going to be able to go through this process of reproduction. And you can see asexual schizogony, so they can clone themselves and make a whole bunch of copies, and then they can go through this sexual reproductive cycle. And here you can see this developing oocyst, and then that is going to be shed in feces. So you are literally pooping out these eggs full of these dormant cells that'll survive in the environment until somebody else comes along and drinks that contaminated water, and then that is going to cause the release of the active part of the life cycle of this protozoan into your intestines, where they can reproduce, because we're effectively little incubators at this stage right here. And if you look in the textbook again, they have another one of those summarizing kind of figures that goes through these distinguishing features with some examples, and then the clinical things, so the kinds of diseases that we can see with some of these different kinds of protozoan organisms that are out there.